Okay students, let's start today's video. Today's video will be on the topic COVID-19 diagnostic testing. In this video we will be covering the basic principle which are being used to test the coronavirus in the suspected patients. Let's begin. So before going into detail of anything, what is the diagnostic testing which we are using for COVID-19, first let me explain you some basic principles which are being used in those testing kits. First one is polymerase chain reaction, or in the short form we call it as PCR. What is a PCR? PCR is a method to analyze a short sequence of DNA or RNA even in samples containing only minute quantities of DNA or RNA. PCR is used to reproduce or amplify selected sections of DNA or RNA. So basically what PCR is polymerase chain reaction. So what is it is doing? It is doing, like for this template DNA, we want this portion, from this portion to this portion, we want multiple copies of from this portion of this portion. Okay, so what will happen? The PCR is a technique which can amplify only this portion. That's why it is known as polymerase chain reaction, क्योंकि एक बार start होने के बाद it is a chain reaction until unless we stop the process. So if I have to explain the process of PCR, then I'll begin from a template DNA. As we all know, this template DNA, it's a double stranded template DNA. One strand is going from 5' to 3' and another strand is going from 3' to 5'. Okay, so first step of a PCR is denaturation. What do you mean by denaturation? Denaturation is basically the separation of two strands. So as you can see in this step, denaturation is going on and the existing two strands get separated into two separate strands. Next step is annealing. What do you mean by annealing? Annealing is basically the attachment of primers. Now if you have seen my previous videos, I have shown you how to design primers. So primer is nothing, basically a small segment of bases based on which DNA polymerase enzyme, which helps in the multiplication of DNA molecule, gets attached, and then the process of polymerization begins. So as you can see here, this yellow portion is a primer and this yellow portion is a primer for this strand. Based on the primer, this annealing temperature can be different, hence the range is given 55 to 70 degrees Celsius. After annealing, this is the green color portion is your Taq polymerase or DNA polymerase. Once the primers get attached, the DNA polymerase now attached to this primer and start synthesizing new strand of DNA on both the strands, both separated strands. Hence this is known as extension. So what will happen after a few minutes, this strand will be completed. And so why it is called extension strand? Because initially it is only a single strand. Now the second strand is being synthesized by DNA polymerase and it is extending, or it is synthesizing the second strand of the template DNA strand to its full form. Okay, so this is basically a small summary of what is PCR. Now moving forward, another concept which you should understand is antibody or immunoglobulins. What are antibodies or immunoglobulins? Antibodies are basically the molecules which are present in human body which fights against the foreign infection. Any foreign infection, our immune cells or our immunity generates antibodies or immunoglobulins which helps in fighting with that foreign particle or foreign infection. So basically this is the structure of an antibody in human body. There are 5 types of antibodies. I've shown here only a structure of single antibody. It is IgG. The 5 antibodies which are found in humans are IgA, IgG, IgM, IgD and IgE. So basically just to cut short this video, I'll explain briefly the structure of this antibody. This lower portion is known as Fc region and it is constant. I mean there will be no changes throughout the human body. This is known as FAB region, and the top version of this FAB region is known as FV region, because this is variable. Why this region is variable? Because try to understand, every infection or every foreign particle will be different from the other. So our immune system have developed a mechanism to cope up with different foreign particles. So whenever a foreign particle comes, it will have a different structure or different set of proteins expressing on its surface. So this variable region of antibody, they will adjust according to the structure of the foreign particle. So that's why it is known as variable region, FV region. Only this region will be different, this region will be different, this region will be different for different foreign particles. Okay, so moving further, now this is the diagnostic process or concept which I want to explain you what is happening on the PCR, or what is happening on the diagnostic kits. So there are two methods which is currently being used. One is RT-PCR based and another is antibody based. So antibody based is very quick. It can give you results within 5 minutes, or maximum it can give you result in 30 minutes, according to the company which is manufacturing it. And RT-PCR basically normally takes 2 to 3 hours to get a result. So what is RT-PCR? RT-PCR is reverse transcription polymerase chain reaction. So what is the difference between a normal PCR and RT-PCR? I will explain in the further slides. Now, how does real-time PCR work with the coronavirus? This I also will be explaining. So moving forward, this is the process of RT-PCR based testing. First step is sample collection. So based on a sample can be anything. For PCR based techniques, a sample can be a blood sample, a sample can be collected from a person's nose or throat. Basically what we want is a genetic material for PCR testing. So genetic material can be isolated from a person's nose or throat, or even from blood. So first step is a sample is collected from part of the body where coronavirus gathers, such as the person's nose or throat. Now second step, what the second step is, remember what we want for PCR. PCR, we want the genetic material as a starting material so that we can begin the process of PCR. So what is the second step is sample treatment. So we remove all the extra material except from the genetic material of the virus or genetic material of the human being for which the sample is being taken. So sample is treated with several chemical solutions that remove substances such as proteins and fats, and extracts only the RNA present in the sample. This extracted RNA is a mix of person's own genetic material and, if present, the coronavirus RNA. Okay. So now we have the genetic material, the starting material from where we can begin the process of PCR, RT-PCR. Now RT is reverse transcription. So next step is reverse transcription. So what do you mean by reverse transcription? Reverse transcription is a process where RNA is reverse transcribed to DNA using a specific enzyme. Remember, during the PCR step in the previous slide, I've told you that DNA polymerase is the enzyme which is used to synthesize the DNA molecule or extend the DNA molecule. Now here we don't have DNA molecule, we have RNA molecule. So DNA polymerase is the enzyme which works only on the DNA. So here we have to convert this RNA into DNA first, then only the process of PCR will be happening. So first step in the RT-PCR process will be the reverse transcription of RNA to DNA, and the enzyme which is doing that thing is reverse transcriptase. Okay, once the DNA is available, then the normal primers will be synthesized. Normal primers will be attached to its 5' and 3' end. So for the 5' ends of the DNA molecule. Then the primers will be attached. Then the DNA polymerase will bind to those primers and extend the DNA molecule, and we will be getting the we will be getting the multiple copies of DNA of this virus. Okay. So now one can ask me, or a general question arises, like from here we have multiple copies of DNA of the virus. So how do we know? What is that? We only got the multiple copies of DNA, right? So how do we confirm that this is the DNA, or how do we confirm that we have got multiple copies of DNA? So for that process, an electrophoresis gel unit is there, or electrophoresis process is there, on which we use to run the DNA molecule. So if we are running a DNA molecule on that electrophoresis gel, basically that gel is made up of agarose. We can made any percentage of agarose based on the DNA size, or we can based on the PCR product size. So based 2% gel can be used, or 4% gel can be used, depending upon the PCR product size. So if we are getting a band in that gel, so that means we have got the multiple copies of DNA, and hence the DNA, multiple copies of DNA, what does it mean? It means the RNA is present, and if RNA is present, it means that person is having a virus infection. Okay. So now the next step would be why we are using, what are the benefits of using RT-PCR. So why we are using real-time RT-PCR? Real-time is nothing. In real-time RT-PCR, what happens is we are getting the result, or we are seeing the result as soon as they are happening. That's it. Nothing more than that. So what are the characteristics, or what are the benefits of using RT-PCR technique for diagnosis of coronavirus? First is it is highly sensitive. Second is it is specific, because if RNA molecule is not there, then the reverse transcriptase molecule will not be able to synthesize DNA, and hence the DNA will not be getting multiplied in the further process. Hence it is very specific. It is giving a reliable diagnosis. It is having a less time, up to 3 hours. It is faster, and it has lower potential of contamination. Okay. So now moving forward, I will explain you what the antibody-based diagnostic kit is, what it is. So basically this is the diagram of an antibody-based kit. As you can see here, this is the process. This is the portion of the strip, or you can say it is like a pregnancy kit, test kit. Here we can drop the sample, blood sample or any sample. Basically, normally we are using blood sample for this. Now, what are these things? CGM or this? So basically, what this composed of? It is composed of this box. I'm talking about this box. So what does it contain? A burgundy color conjugate pad containing SARS-CoV recombinant antigens coated with colloidal gold SARS-CoV-2 conjugates and rabbit IgG gold conjugates. Basically what it contains is SARS-CoV-2 recombinant antigens. Remember, see, COVID-19 is a virus. Okay, or SARS-CoV-2 is a virus. COVID-19 is a disease. So SARS-CoV-2 is a virus. So once it enters the human body will synthesize certain antibodies with respect to fight this infection. So these two antibodies are IgG and IgM. Okay. So these two lines G and M are the anti-human IgG. This is anti-human IgG and this is anti-human IgM. So try to understand the concept here. Here what is happening is binding process it is happening, or you can call the antigen antibody interaction is happening. Human body is having antibody against the virus infection, and here we have lined the counterpart of those antibodies, IgG, anti-human IgM and anti-human IgG. Okay, so if a virus infection is coming and it will be having, if it is having IgG antibody synthesized with response to SARS-CoV-2 recombinant, with response to SARS-CoV-2 infection, so those antibody will bind to this anti-IgG portion, and if the patient is having antibodies IgM with in response to the infection of SARS-CoV-2, then those antibodies will bind to anti-human IgM. Okay, once they bind, they give a burgundy type of color. Before going into this binding process, what this portion is contained, it contains a nitrocellulose membrane containing IgG line coated with anti-human IgG, okay, and anti-human IgM, and a control line coated with goat anti-rabbit IgG. And the most important point which this portion contains is the SARS-CoV-2 recombinant antigens conjugated with colloidal gold and rabbit IgG gold conjugate. So basically this portion is having antigens, SARS-CoV-2 antigens, okay, and antigen will bind to antibodies, corresponding antibodies. So this whole strip is composed of SARS-CoV-2 recombinant antigens, which are further conjugated with colloidal gold to increase the specificity, and of course rabbit IgG gold conjugates. Now when the virus infection, if a person is having virus infection, so certain antibodies will be synthesized in response to that virus infection. If the person is having synthesized IgG antibody, then those antibodies will bind to this portion, anti-human IgG lines, and if the person is having synthesized IgM, so those antibody will bind to anti-human IgM. So that way, after binding, they give a burgundy type of color. Now what is this control line? Control line is composed of goat anti-rabbit IgG. Basically what does this control means? If it is there to check if the strip is okay or not. If while testing this burgundy color is not being present, then the test is not valid. If it is present, then the test is valid. It might be the case that person is only having a single antibody, like some person can only generate IgG, or some person can only generate IgM antibodies, or some person can generate both IgG and IgM. So some person will be getting result like red color, a burgundy color in C and burgundy color in G, and some persons can have a result like burgundy color in C and burgundy color in M, and some other person can also have burgundy color in C, burgundy color in G, and burgundy color in M. So this is the rapid action kit principle which is being utilized nowadays. The company Calcex has developed this apparatus. It is FDA approved. So that's it. This is the diagnostic kits which are not now available in the market, and people are being tested with these kits. One more thing, this process, this rapid action kit is not 100% guarantee, because it differs person to person to show the presence of antibodies after the infection of coronavirus. So it is not 100%. So doctor prescribed RT-PCR based technique if the person is being tested negative in this. Okay, so that's it. Goodbye for now.